So as I'm sure you know, there are generally two strands of DNA and they come in base pairs. So some of these bases only pair with other bases. And really, it's all about hydrogen bonding. So that's where the chemistry comes in. And biology is just basically zoomed out chemistry, which is then zoomed out physics and blah, 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 so on and so forth. So you can see here, I'm just drawing you a little picture, right? Here's A and T. And you can see why they match up. They have two hydrogen bonds, whereas G and C have three hydrogen bonds. So that helps them keep from mismatching. You can see that we have hydrogen bond acceptors. Right, so they don't have the hydrogen, they have the lone pair that coordinates with that hydrogen. And then we have hydrogen pair or hydrogen bond donors. They have that hydrogen bond, right? That NH-OH bond. In this case, they're just NH bonds. And then same thing here, right? So here are our acceptors. And then there are our donors. And you can see one set has two, the other set has three, so that helps them from mismatching. Here's just another picture of this DNA molecule, right? We can see it gets big, looks cool, right? All that kind of good stuff. One cool thing about the DNA bases is that they're all aromatic, which helps them be very stable, but it also makes them very planar. So remember when we learned about aromaticity, one of the requirements is that it's planar. And we can see that our DNA bases here are indeed planar. Note that then this leaves a bit of a gap in between here, in between your DNA bases, and other aromatic molecules can actually get in there. So other aromatic compounds can, it's called intercalate. Basically just means get in between there. And then that messes with the cell, messes with the DNA, and can be a cause of cancer, which cancer is just basically uncontrolled cell reproduction. And so that's is one of the causes, right? Causes damage to the DNA. The cell then just keeps reproducing, and then you end up with a tumor. So one way that we come in contact with a lot of these aromatic compounds is through cooking our food, especially when you grill. So when you grill food, you end up with that, kind of nice, tasty char. Unfortunately, that char is basically only partially burnt hydrocarbons, and they often end up in very stable positions. And think about stable molecules, those are our aromatic hydrocarbons. So there's just one example of them, right? This is our PAH, our polycyclic aromatic hydrocarbon. Above is just an example, right? But we formed all of them. So whenever you grill food or really do any kind of cooking, mostly grilling though, you're going to end up with these polycyclic aromatic hydrocarbons. You eat them and they could end up intercalating your cell, which could end up causing cancer. I haven't done much research in biochemistry, but I did do a little bit during my undergraduate. So here's a poster of me during my undergraduate. You can see I went to school in central Pennsylvania called Shippensburg University. And I did the DNA binding studies of aryl sulfoxides. And so we used this intercalation mechanism to our advantage. So what we did was we synthesized some photoactive radicals, which are those unpaired electrons. And you can see them right here. There's an example of two of them. And then we basically did a three-step procedure to study how they interact with DNA. So first we'd interact them with DNA and they intercalate. Step two, we shine the light on them, right? That's what photoactive means. And then they'll produce a radical. Once you shine the light on them, the radical will then cut the DNA and you'll destroy it, the DNA, and thus the cell, right? With the idea being that you can kill cancer cells that way. And there's a lot of other cancer research. The main issue with cancer research is that a cancerous cell and a regular human cell, they're both your cell. And so they look very much like one another, right? So the main question that when you study types of cancer cures is you have to ask yourself is how do you make a drug selectively go into cancer cells versus a healthy cell. So we've got all kinds of drugs that'll kill cancer cells, no problem. The problem is they'll also kill your healthy cells. And so that's a big question that needs a lot more research. So if that's something you're interested in, go ahead and go study some more biochemistry and find us the cure.